May I start, sir?
Yeah, please.
A very good evening, respected director, sir, faculty members, dear student and audience. I, Dr. Sant Kumar Verma, Associate Professor, ISF College of Pharmacy, Moga, would like to welcome Dr. Himanshu Pandey, Deputy Registrar, Central Institute of Higher Tibetan Studies, Sarnath, Varanasi, India, at the online platform of ISF College of Pharmacy, Moga, for delivering expert talk on nanoparticulate-based ocular delivery system for controlled drug delivery to the anterior segment of eyes. I request to respected director, sir, please welcome our speaker, Dr. Himanshu Pandey, sir. Over to director, sir.
Thank you very much, Sant Kumar Verma ji. So, on behalf of ISF Dialogue Series and ISF College of Pharmacy, myself, Dr. G.D. Gupta, welcome Dr. Himanshu Pandey ji from core of my heart. And sir, this dialogue series is the under the ages of we are going to running IQAC, and the purpose behind of this series to invite some of the dedicated, dynamic, and young scientists and researchers, those who are doing the work in the special field, so that we can provide a common platform to all the professionals. Sir, I inform to you that we have invited from the abroad number of the scientists from the NIPER, central universities, and some of from the industry peoples also. So I think this is the 78th of the dialogue series today. So I know that your busy schedule, and I am very very thankful to Dr. Himanshu Pandey to accept our invitation and provide the very crucial or potent time for the delivery of the lecture. So again, I welcome you, sir, in this ISF dialogue series, and I am thankful Dr. Sant Kumar Verma to provide the opportunity to me to interact such types of the dynamic and down to earth person, Dr. Himanshu Pandey. And I also request to all of the audience, if you have any questions or any doubt, you can send to the chat box, and our research team will go together and we will try to give the justify your queries, and we can also connect to the speaker if required to the our IQAC and scientific team. So over to Dr. Sant Kumar Verma.
Thank you, respected director, sir, for briefly elaborating about our dialogue series. Now, I would like to invite my mentor, Himanshu Pandey, sir, for his lecture. Over to sir.
Thank you, Dr. Sant Kumar Verma. First of all, I would like to thanks the management of ISFF College Moga, particularly the Department of Pharmaceutical Science professionals, for providing me the opportunity to speak on the topic of nanoparticular-based ocular delivery system for the delivery to the anterior segment of eye. And particularly, I thanks to Sri Praveen Garji, the chairman of ISF College Moga, Professor G.D. Gupta ji, for inviting me. And I am thankful to Dr. Sant Kumar Verma, who spoken to me 2-3 weeks before that he requested that I have to present some recent findings of my research. So I am happy to share my views on this particular topic. So shall I start, Dr. Sant?
Yes, sir.
Yes, sir.
Sure, sir.
Okay. So good evening to everybody. Myself, Dr. Himanshu Pandey. Presently, I am working as a director of CT at Central Institute of Higher Tibetan Studies. This institute is located in Sarnath of Varanasi district. And today, my topic for the presentation is nanoparticulate ophthalmic system for controlled drug delivery system to the anterior segment of eye. Actually, this is the research study, and in 30 minutes it is not very possible to elaborate each and everything, but I will try to my best to give the all the crucial aspect of my research work. So before going to the formulation part, I would like to say that I would like to brief about the physiology of and the anatomy of the eye. Actually, the human eye is divided basically into two parts, that is the anterior segment and the posterior segment. Anterior segment consists of cornea, conjunctiva, sclera, anterior uvea, and aqueous humor, while the posterior segment of eye comprises of retina, pupil, vitreous humor. And the disease or the disorder which is related to the anterior segment of eye are particularly the infectious disease like the conjunctivitis, scleritis, keratitis, uveitis, retinitis, and the glaucoma. While the disease which is associated with the posterior segment of eye is the retinitis, and particularly nowadays, India is becoming the capital of the diabetes. So diabetic retinopathy is the major disorder which is very crucial nowadays for those patients who are diabetic. So these are the disease and the disorder which is associated with the eye. But if we talk about the uh, treatment or the therapeutics of the eye, about 90% of the drugs which is used for the treatment of eye disease or disorders comes in the form of the eye drop. 90% out of the total formulation of the ocular, 90% of the formulation is in the form of the eye drop. Now what's happened, whenever a people you might have experienced that if we instill 1 or 2 drop of the drugs from the eye drop, number of drops comes out from our eyes, it will drain. So if we had instilled only 2 drops, but there are a huge amount of the tears which comes. Why it's so? Because whenever we put the eye drops comes into the contact of the eye, our body, our eye detect it as a foreign particles, foreign bodies, and to remove that particular foreign bodies which actually contains the drug, they start releasing, the lacrimal gland get activated, and they start producing the tears more and more, and the drugs get drained out. The drugs get drained out. This is not a only one parameter by which the drug get drained out. There are the number of parameters which I am going to discuss in the next slides. So apart from the drainage by the tears production, what happens, if in the open eye only 30 microliters of the fluid can get accumulated, only 30 microliter. So if we instill 1 or 2 drop of the drugs from the eye drop, our lacrimal gland get activated, which detect these drops as a foreign particles, and simultaneously our lacrimal gland get activated and tears production starts. So the total accommodation volume of the fluid in the open eye is only 30 microliter. But when the lacrimal gland activated and tears production start, the drugs, all the fluid get drained out from the eye. That is the major loss. Second loss, when we blink our eye, when we blink our eyes, the rest of our volume of the drug also get drained out. Now, apart from that, there are the various other factors like there one nasolacrimal duct is available in our body. So through these nasolacrimal duct, the drug get drained out and comes to the mouth. You might have feel the bitterness of the eye drops when you instill some bitter drug in the eye. You will feel its taste in your mouth because from the upper palate, this nasolacrimal duct get open to the upper palate of the mouth, and the drugs comes through these route into the mouth. This is the again one part by which the drug get drained out. So by blinking, by production of tears through the nasolacrimal drug, even the rest of amount drugs which is available for the action, there are the various barriers are there in our anterior segment of the eye which restrict the drug to get enters into the target section. So by the study, it has been observed only 5% or less than 5% of the drug which is instilled into the eye through eye drop gets available for the biological activity. So in one sentence, we can say that bioavailability of the drugs which is instilled through eye drop is less than 5%. Next. So before getting into the nano formulation, because this nano was a buzzword during the era of the 2001 to 2010, but nowadays a lot of work has been done on this nanotechnology field. All of you might have been knowing that what is the nano and what is its dimension. But one thing I want to say, that there are the two not branch, but the two terminology, that is the nanoscience and nanotechnology. Student mean they always get confused that what is the nanoscience and what is the nanotechnology. Are these two are the same thing or the different thing? But the study of the particles of the nano dimension is the nanoscience, and whenever we are using that nanoparticle for certain type of application, this area comes into the nanotechnology. And Richard Feynman was a physicist who considered as the father of the nanotechnology, Richard Feynman. Now, the question is that what is the advantage of the nanoparticle and why nanoscales are widely used nowadays for the biological application? Because when the particles get reduced into the range of the nanometer, say like less than 100 nanometers, their property get changed, like surface related property and the quantum property get changed, and which leads to the change in the surface volume ratio, then optical property of the materials, their thermal conductivity, melting point, solubility, and so on. Even the rheologic property also get changed. So same gold particles, we know that the gold appears to be yellow, but when we reduce the size of the gold particles below 50 nanometers, it appears into red. And when it comes below the 10 nanometers, the same gold appears as a black. So by reducing the size of the particles into the nano range, the properties get changed. So you might have seen the colored glass in the Greek Romans era. The Greek Romans in the era, they usually paint their window glass in the different color. But you will be surprised to know that the same materials were used to color the glass into different color, because by changing their particle size, the same materials appears in different color. So that means that nanotechnology and these type of things are not very new, because as we had said that the Richard Feynman, the father of the nanotechnology, no, beyond that, before that also, in Hindu calendar also, in our culture also, we are using the bhasma, swarn bhasma, silver bhasma, and all these heavy metal bhasma in our Ayurveda formulations. So we are using the nanoparticle from those days. Now, next, if we talk about the controlled drug delivery system, it do had its own advantages, because we know that by using the conventional system of the drug, you can say, I am showing the graph of the plasma drug concentration versus time. You can see the graph of the seesaw graph of the means the representation of the multiple or the conventional delivery system. So there is a chance of the drug to get approach the toxic level, and sometimes it is beyond the subtherapeutic level. So if we discuss about the prolonged drug delivery system, under prolonged drug delivery system, there are the two systems basically, that the sustained release system and controlled release system. In sustained release system, you can say it simply extend the release of the drugs up to the certain period of time. Sustained drug delivery simply extend the release of the drug up to the certain period of time. But if we talk about the controlled drug delivery system, it can be defined as it extend the release of the drug up to a certain period of the time with the predetermined rate. Means in controlled release system, the release is with predetermined rate, while in sustained release it is not like so. It simply extend the release of the drug. So particularly in the controlled drug delivery system, it follows the zero order kinetics. And next slide, the advantages which is associated with these extended release formulation is the total dose is low. Means the if we say that the total dose of the domperidone is conventional dose is 10 mg, and it has to be taken every 6 hours, then it comes about 40 mg of the drug is required for a day. But if we convert into a once a day formulation in the controlled release or sustained release formulation, we doesn't require 40 mg of the drug to get action for 24 hours. The dose gets reduced. There is the formula. I am not going to discuss here. So the dose get reduced about 24-25 mg will be used for getting the same action for that particular for a day. So another is to reduce the undesirable side effect of the drug, definitely, because we had seen in the previous graph that drug in the conventional system may reach to the toxic level, or may there will be beyond the less therapeutic level, that is insert therapeutic level. And yes, that it reduced the dosing frequency, because if you you have to take the 4 drug for 4 times a day for getting action for 24 hours, and if you convert into sustained or controlled release, you required only 1 tablet for a day. So dosing frequency definitely get reduced. Better patient acceptance and compliance, of course. Less fluctuation at plasma level, we have seen in the previous graph. More uniform drug effect. Previous graph we had seen that the in controlled release, the rate of the drug is released with the predetermined rate, and definitely it will lead to the improved efficacy and safety ratio. But still having the different various advantages, some disadvantages is also associated with the controlled drugs delivery system, or you can say the extended drug delivery system. That dose dumping is one of the major drawback, because if the formulation is not good enough, the total amount of the drug which has been loaded into the formulation get exposed. It get exposed, and the total dose get exposed, and it leads to the toxic level when the total drug, because we had formulated the formulation in such a way that it will release slowly and slowly, and if it will release slowly and slowly, definitely it is going to be absorbed slowly and slowly, and the plasma concentration will get maintained. But if abruptly, if due to certain region, if the drug which is loaded into the formulation get exposed immediately, then it will shoot to the toxic level. So dose dumping is the major disadvantages of the which is associated with the prolonged drug delivery system. And another disadvantage, need additional patient counseling, because in certain type of tablet, suppose it is mucoadhesive or bioadhesive tablets are there, matrix tablets are there. So sometimes patient used to break the tablet. Suppose if he feels that I need only half tablet, what he will do, he will break the tablet and eat. By breaking the tablet, the coating, the polymeric coating get distorted, and there will be the again the means the bad effect of that particular drug. And in certain type of formulation, it has been seen there is stability problem, and while using the various polymers, specific polymer, they may increase the cost of the certain type of the such formulation, prolonged release or sustained release. So these are the disadvantages which is associated with the system. Now now let's come to our main part, that is the preparation of the gentamicin sulfate loaded nanoparticle for the treatment of the infection of the eye, which is particularly infection of the anterior segment of eye. We had used our model drug gentamicin sulfate as an antibiotic, and we had prepared the formulation, which we used the nanoemulsification method. Actually, we had used the 2 method, that is the nanoprecipitation method, which I am not going to discuss here, and another is the nanoemulsification method. Here I am going to discuss about this particular method, nanoemulsification method. In this case, we had used the aqueous solution of the gentamicin, 5% weight by volume, and we had emulsified it in the solution of the methylene chloride containing Udragid polymer. Udragid RSRL 100 grade polymer were used, which is particularly used to control the release of the drug. So here we had used the Udragid polymer, and this water in oil emulsion was prepared by the process of the emulsification method, and Then after getting the water in oil emulsion, again the emulsification was done in the 1% polyvinyl alcohol solution. Polyvinyl alcohol is nothing but it is a stabilizer. It was used, and once the water in water emulsion get prepared, the whole system was homogenized at 60 bar for 3 cycle. And lastly, the micro emulsion or the nano emulsion, you can say we get water in oil in water. We again drop by drop, we again put up into the 100% of the polyvinyl alcohol solution for stability purpose. And for a whole night, we stir slowly and slowly for the evaporation of the internal phase. That is the organic phase in which the polymer get dissolved. Once the internal phase get evaporated, the polymer gets solidified, and the polymeric nanoparticles you will get the polymeric nanoparticles. And by adding the cryoprotectant and lyoprotectant, the this formulation get lyophilized, and you can see the figure 1, the gentamicin solution 1A, and 1B is the gentamicin nano suspension, while in figure 2 you will find the lyophilized gentamicin loaded nanoparticles. So these emulsification method were used. Next, after getting the nanoparticle, we had characterized the nanoparticles for the surface characterization structural, that is the scanning electron microscope SCM, transmission electron microscope TEM, and AFM atomic force microscopy was used. You can see the figure of the different. The particles are very spherical in shape, and as the SEM image depict, and in SEM image also you can find the particle size is more uniform. Later on you will see the polydispersity index value, that it will signifies the means the uniformity in the particle size. Later, for the crystalline status, we had performed thermogravimetric analysis, differential scanning, colorimetry, and X-ray diffraction. And apart from that, we had performed the zeta potential, polydispersity index, and the surface charge. Next, in thermogravimetric analysis, it is particularly used to measure the amount and the rate of change in the weight of the material as a function of temperature in the controlled environment. Now, by the graph, you can see that the green lines is of gentamicin sulfate alone. The red line comprises of udrazid RL, RS100 and gentamicin sulfate, that is the nanoparticle. And the blue lines is the of pure udrazid RS, RL100. Now, you can see that this gentamicin green line, there is the sharp degradation is observed at 2 phase, that it is near about 230-degree temperature, and second one is near about 300-degree temperature. And in blue line, udrazid sulfate, steady decrease in stability is observed with the temperature, while in the case of the nanoparticles, which is contain gentamicin and udrazid, you will find the drug is stable till 225 or 220 degrees centigrade, although there is a sharp degradation beyond 230 to 40 degree, but we can say in comparison to the pure drug and the pure polymer, our composite is quite stable till 220 degrees centigrade. So that is the one of the means the figure which we had seen analysis. Second one is the differential scanning colorimetry, and this analysis is used to characterize the thermal behavior of the polymer udrazid RR100 and the drug gentamicin sulfate, and as well as the polymeric nanoparticle which contains the drug. Now you can see the two sharp endotherm were obtained with the drugs gentamicin sulfate, that is near about 160 degrees centigrade, and another is about 320 degrees centigrade. Moreover, in the case of the physical mixture of the udrazid RS RL100 and gentamicin sulfate, there was increase in the glass transition temperature near about at 240 degrees centigrade. These results suggest that the dense morphology and the higher degree of the crystallinity of the polymer blend can thereby be considered as the improvement of storage stability of the system. Next, we had calculated the encapsulation efficiency of the drug, and this can be determined by, it is the ratio of the total gentamicin which is used for preparing minus free gentamicin about upon total gentamicin into 100. So what is the free gentamicin? Means the total gentamicin which is used for the incorporation into the polymeric nanoparticle, where here we had used hydrogen polymer. After encapsulation, what we do, we take the about 5 ml of the overall content, and after centrifugation, high centrifugation at 15,000 to 20,000 rpm for the 20 minutes, we take the only supernatant, means the nanoparticle gets settled down. Nanoparticle is the hydrogen nanoparticle which encapsulate the drug. It gets settled down, and we only take the supernatant, and we take the OD of that particular supernatant for determining the free amount of gentamicin into the system. So after getting the free amount of gentamicin minus the total gentamicin which is used upon the total gentamicin into 100, we will get the encapsulation efficiency. Now we had prepared the 5 formulation, as you can see into the this table. This is GN1, GN2, GN3, GN4, and GN5. The drug polymer ratio is presented here. You can find the particle size also. The minimum particle size was near about average was 145 nanometer, and the biggest one was GN5, that is 243. Average particle size was 243 nanometers. And the zeta potential, you can find it is between 20 to 30, and polydispersity index, as you see, that is 0.2, 0.3, 0.3. The polydispersity index value is not very good, but it's okay, that we can say our formulation, the particles which is formulated, it was the size of the particle was nearly uniform. Because what happens, merely by determining the average particle size, we can't say that our formulation is very good and it is called as a nanoparticle. Because suppose if we take 3 particles, one particle is of 200 nanometer, and another is of 400 nanometer, another is of 600 nanometer, then the average will be 400 nanometer. But we know that 600 nanometer particle is also available in the system. So how can we say that the size of the particle which has been formulated, which has been prepared, is uniform in size? The polydispersity index will gives the answer. Okay. Now second, again we can see the encapsulation efficiency. You can see for formulation number GN1, the encapsulation efficiency is comparatively good. That is, it is 81.79, and the release efficiency for 12-hour study, about 92% of the drug get released from the encapsulated system. Next. In vitro release study, similarly we had performed the in vitro release study, and here we had used the dialysis membrane with the molecular weight cutoff was about 12,000 to 14,000 Dalton, and we had take the drugs 5 ml gentamicin sulfate suspension, and we had performed, and we had for the dissolution media we had used the simulated Pierce fluid. So the you can see with the graph of GN1, the formulation number GN1, you can see about 90-93% of the drug is released up to 12 hours. So and the release, the pattern of the release of the drug was nearly uniform. You can find with this graph that we can say that the release was controlled, and it is released slowly and slowly, up to 24 hour and 24 is up to 12 hour, and more than 90% of the drug get released. Now, as far as the release kinetics is concerned, we can see this table, and we had plotted the value into the four models, that is zero order kinetics, first order kinetics, Force-Meyer-Pipaz equation, and Higuchi one. So the release of the gentamicin sulfate from the polymeric matrix is through Fickian diffusion, because the value we can say the regression R2 value of the Force-Meyer-Pipaz equation, it is 0.99, 0.98, 0.99. Whether in Higuchi it is less than 0.99, it is 0.97, 0.97, 0.96. Whereas in zero order it is again 0.96, 0.95. First order it is again very less. So we can say that the values properly fit into this Force-Meyer-Pipaz model, and the n values was found to be 0.44, 0.11, 0.43, and it means that the release of the drug from these polymeric nanoparticle is through Fickian diffusion. Means the no erosion is taking place, the polymer, the nanoparticle remains intact, and the drug release from that particular nanoparticle slowly and slowly through the Fickian diffusion. Now again, if we talk about the stability studies, we can find after storage for the 6 month, we had used the formulation code, the optimized formulation. GN1 is the initial, and the GN double plus as a freeze-dried nanoparticles which is used. Initial values of the particle was 145 and another one is 152 nanometers. After 6 months at 5 degrees centigrade, the particle size slowly slightly get increased, and after 6 months as 25 degrees centigrade, 60% relative humidity, again there was a slight change, insignificant change was there. And if we say, so we can say that as far as the size of the particle is concerned, there was no vast means changes appear. We can say only 5 to 10% of the size get increased. The increment in the size was 5 to 10%, while the drug loading percentage, you can find the initial values was 81.79 average, while after 6 months it was 78 and 78, means that some drug get degraded with due course of time, but it was not very significant. So we can say that this GN1 formulation is stable also. Now we had also performed the in vivo study in the rabbit. So first of all, we had test the ocular irritation of the nanoparticle, whether the nanoparticle is irritable or not, it is compatible with the patient or not. After getting animal experiment in the rabbit, we find, because the normal winking rate of the eyelid is 1 to 2 winking per minute, means the human or the animal blink 1 or 2 times in a minute. So for group 1, we had used the control simulated tears fluid of PS 7.4. For group 2, we had used the commercial eye drop of the gentamicin sulfate. And for group 3, we had used the nano suspension. So winking count for the control was 8, average. Commercial eye drop, it was 7.33. And for the nano suspension, it was 10. Although it was 10, but it is within the limit, within the acceptable limit. So we can say that our formulation passed the ocular irritation test. Simultaneously, we had performed the antibacterial test also in the viability test. So viability test, you can see into the table that the Cmax is 14.95 and for the nano suspension, and 7.98 for the gentamicin sulfate solution. So you can see the Cmax, the Tmax, and particularly the viability. If we talk about the viability, then you can see the area under curve. The area under curve for the gentamicin nanosuspension was 87.34, the huge viability, as compared to the simple gentamicin sulfate solution, which was quite less, that is 38.14. Sorry, I had forget to change this slide. This is the in vivo release profile. And next is the in vivo antibacterial activity. We had used different groups, control groups, where there was no drug treatment. Group 2, rabbit treated with the gentamicin sulfate solution, and group 3, rabbit treated with the nano formulation. You can see with the figure in the control, where the no drug treatment was given for the infection with the infectious eye. So with means day to day, the condition of the eye get worse. For the control, you can see after the 6th day, the condition of the eye get worse. While in the case where only gentamicin solution was used for the treatment of the infection, you can see that up to the day 6, the eye was not in proper condition. There was not permanent cure of the infection of the eye. But where the nano suspension was used, you can see that at day 6, the eye get particularly cured. The fresh eye, the uninfectious eye, you can see in this figure. Now, what is the significant in the nutshell, that with these formulation, nano formulation, the overall improved the pharmacokinetics and biodistribution of the therapeutic agent to the intended site. It reduced ocular toxicity as a consequences of the preferential accumulation at the target site. It reduced dosing frequency, as the present research is focused to achieve a once-a-day daily formulation. Prolonged therapeutic effect because the controlled drug delivery of the drug from the carrier. It lowered the dose required for efficacy. It particularly increased the stability of the formulation, and it focused on the possible economical production of the large scale to increase the stability of the developed system during storage. So these was my findings. Thank you so much. Any question or anything, Sant, if you would like to ask, or someone want to ask, you can ask.
Yes, sir. Yes, sir. Now, I would like to invite IQAC coordinator, Professor Siddharth Mehan, to pay vote of thanks to our distinguished speaker. Over to Dr. Siddharth Mehan. Sir, please unmute yourself. Sir, please unmute yourself.
Myself?
Am I audible, sir?
Yes, 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 sir.
Yes, sir. Thank you, sir. Thank you, Dr. Sun. And we are very thankful and highly appreciated today's our renowned guest and speaker, Dr. Himanshu Pandey, Deputy Registrar, Central Institute of Higher Tibetan Studies, Sarnath, Varanasi, India. And we are very thankful to the concise knowledge about the nanoparticles ophthalmic system for controlled drug delivery to the anterior segment of eye. And the work presented by the Dr. Himanshu Pandey was excellent and very important. And again, over to Dr. Sant Verma.
Thank you, Dr. Siddharth Mehan, for thanks to our guest of speaker. Now I would like to invite Director Sir to speak few words. Now I would like to invite Director Sir. Sir, please unmute yourself.
Thank you very much, sir. So very nice lecture, and you have given very precise and important information. So I hope that, as you know, that most of the institutes, they are the working on the related to the skin, related to the oral delivery system, topical formulation. Only few institutes they have established the lab related to the ophthalmic. and they are the working on the eye and related to the eye disease. So even we are also interested to establish here one of the laboratory, so that we can also work on the some of the ophthalmic product and related to the disease of the ophthalmic. So you have given the very nice, and I have come to know some that you have used some of the eye cornea and the eye parts for the various of the studies using the animal. As we know that nowadays when we perform the animal study related to the rat, mice, guinea pig, rabbit, for this purpose need a very strong protocol. What if the person want to do such types of the activity? It is very important. I know that one of the person and Dipsaru, and he has throughout the life he did the work on the ophthalmic. So obviously this is the one of the excellent field, and nowadays this is the required, because most of the peoples they are the using the specs, number of the, you can say, infective disease, there is the problem of the vision. So why not we can develop some of the dosage form which will be the useful for the long sighted of the action and the protection of the eye, because one of the external internal organ is the one of the highly sophisticated required sterile preparation. So this is the excellent, because I am the background of the pharmaceutic. So even that we are the thinking about that, ki why not we there develop one of the section of the ophthalmic drug delivery system. So our team will be the connect to you, and I want to the establish here one of the small section, because we have the zebrafish lab, we have the excellent animal house with the breeding and trading facility, cell line culture, and the microbial cell culture, tissue culture lab, all types of the lab most probably we have. What I feel that this is the space required in my institute, so that we can do the work in all of the doses form. So as and when you have the time, and you can visit this institute, and we can do some of the collaborative work for the research, and we can establish some of the commercial preparation. So I want to over here mention that in those days our team is the running 6 of the MSME project, and I feel that all MSME project we want to connect with the transfer technology, and some of the project I want to the transfer to the technology to the industry also. So one of them I want to include one of the our pharmacology person, they want to do the work on the ophthalmic system, but due to some of the, you can say, facilities, because need some of the sophisticated facilities and proper labs. So now our management is the agree to develop the one of the excellent labs. So thank you very much, sir, for your precious time, and you have elaborated very technical manner. So I hope that your lecture it will be the very helpful to the each and every of the pharmaceutical person, and those who want to work on this field, they will be take the benefit of this lecture. So thank you very much, and thanks to the all coordinator of this program, Dr. Sant, Dr. Siddharth Manji, and I am also thankful to the management to provide such types of the opportunity to continue this ISF lecture series from 101 hours, so that we can provide some of the good knowledge, good expert talk to the society. Thank you very much, sir.
Thank you, sir. I want to add one thing, sir. Actually, we had received one project from the Department of Biotechnology, New Delhi. We are working on the diabetic retinopathy delivery of the drug through ocular eye drop, because it is the disease which is associated with the posterior segment of eye. And merely by using the eye drop, we can't transport the drug from the anterior segment to the posterior segment. For that, we are using the cell penetrating peptide as a carrier to take the drug from the anterior to the posterior segment to the target site. But beyond that, I want to tell you that, sir, we are the Tibetan University, and we are the huge Tibetan medicine faculty. We are using formulating here the Tibetan medicine formulation, and we are having the full-fledged faculty of the Tibetan medicine. So if you want to have a collaborative research of by using this Tibetan medicine, because it is very prominent and particular for the means the metabolic disorder disease, whether it is diabetic or arthritis, the people are getting very benefits by using these medicines, because we are procuring the crude drugs from the high latitude of the Himalayan range. So I request you to send your team, or in person you can visit here, in our Tibetan medicine faculty, and we can jointly write a project, and we can do some research on these unexplored area, because this is not very scientifically explored. This is very conventional and traditional medical system of the Tibet, but we can explore scientifically in these area also. So I request you, whenever you feel have a time, please do visit to our Sarnath in our institute, so that we can find a way to get collaborated and have a research on these Tibetan medicine.
Thank you very much, sir. So very nice. You have given the information, and obviously we need to work on the out of box. And Tibetan medicine is one of the great field and need to the job over here. So I just definitely to interact with you telephonically as well as the email. So we will 2-3 person they can visit your institute, and then we can go through what types of the facility we have over here. We can do some of the collaborative activity as well as the MOU with your university and college. Thank you very much, sir.
Thank you, sir. Thank you so much. Thank you, son. Thank you, team.
Thank you, sir. Thank you,
Thank
sir.
you, sir. Thank you, sir. Thank you very much, sir.